because I know that we have some English speaking amongst our audience. And I hope I recognize some of the faces from the exhibition opening yesterday. And for those of you who has not seen the article biennial yet, you have still some time because it's going to last until October 2nd. So I hope to see you there at the exhibition. And as part of our article program, we're going to have art, science, and cocktails tonight. And we've invited scientists and artists. We have 4 scientists with us today and 2 artist presentations. And we also have a fabulous cocktail menu that's been developed here of the staff here at Sting. So we hope you'll test it out and enjoy it. And we're going to give you some time to top up your drinks. So we're going to have 2 presentations and then a short break. And if you have any questions, we have time for a really short Q&A session after each presentation. So first presenter up is Dr. Andrea Baggi. She's the professor in environmental engineering of the Department of Mathematics and Natural Science at Stavanger University. She will be presenting Human Ecosystem, Us and Our Microbes. Welcome, Andrea.
Thank you, Hege. Hi, everyone. Good evening. My name is Andrea Baggi, and I am a microbial ecologist. And today I would like to share some information with you about your microorganisms, the things that live on and inside your body. I'm waiting for the first slide. Okay, all right. So I hope everyone can see this. Well, my original plan was to start with a personal story about my dog, but I think if I started on that, then I would definitely go over the 15 minutes. So if you're curious, you can ask in the break, and I'll just very quickly move on to the next. And start with an observation. Maybe I'll just move over here. To me, it seems that we live in a world these days where bacteria are not very welcome. We look at them as those germs, disease-causing agents that we should really just try to get rid of, wash them away, or kill them with antibiotics. This makes a microbial ecologist very sad. Those bacteria that cause diseases, they are called pathogens, and they are really just a small part of the entire bacterial diversity that is out there. So if you ask a microbial ecologist, they will immediately tell you that, oh, bacteria are fantastic. They are actually running the entire planetary ecosystem. Basically, you should start thinking about microorganisms as the engine in your car. You don't really see it necessarily until something goes wrong, but it still runs the car. We also don't see microbes, but they do a lot of important things. In the ecosystems that we see, like the forest, the soil, and the ocean, but also, apparently, in the human body. And this is what I would like to talk about today. Because approximately 10-15 years ago, microbiologists made an astonishing discovery. They found out that our human body contains approximately 10 times more microbial cells than human cells. I hope that this shocks you a little bit, at least. This basically means that we are more of a microbe than a human. And the only reason we don't look like a microbe is because they are so much more smaller than human cells. So this is how the entire story of the human microbiome and this human ecosystem began. Now we know that there are hundreds or thousands of different bacterial species on our skin, in our mouth, in our lungs, or even in our stomach. And they form distinct communities in each body part. So if you look at your elbow microbial community, it's going to be very different from the one that is in your mouth. because there are very different conditions on your elbow and in your mouth, obviously. But the largest diversity of bacteria can be found in our gut, in our large and small intestines. And this is the microbial community, the gut microbiota, that I would like to talk a little bit more about today. So now that you know that there are so many microbial cells in your body, you may be wondering, all right, but what are they doing there? Why are they there? How did they get there? Do we really need them? And do they in any way impact our health and well-being? Is there something like the healthiest possible gut microbiota? Now we'll start by answering the last question. We don't know. The only thing we know is that perhaps the more different species, the highest diversity you have in your gut, the better it is for your health. So hopefully by the end of this talk, you will agree with me that in some cases, it is much better to try to obtain as many bacteria as possible rather than trying to get rid of them, especially when you are young, especially when you're young. So I will tell you a little bit about what these bacteria do in our guts. And, well, bacteria in our guts do what bacteria do the best. They basically transform chemicals. They are like little machines that turn one chemical into another. They use it as a food, energy, and they build their own cell material from it. So basically, they feast on the leftovers of your food that you just had for dinner, the things that your human enzymes and human cells were not able to consume. It will all go for your bacteria. Now this is great because we want to maintain them. And here I would like to mention that different types of bacteria like different kinds of food, just like different people. So there are those types that prefer leftovers from a meat, or there are bacteria that would prefer the fibers from your vegetables. And yet again, there is another group who would really like you to have some cheese for dinner. So I would say that if you agree with me and try to obtain the highest diversity in your gut, then you should eat all the different kinds of foods available on a regular basis. Now, let's look at another way, another thing that bacteria in our gut can do. Basically, they can extract more energy from the leftovers of our food and then give it back to us. So basically, they can help us digest the food better and get more out of it. And in this way, actually, they have a say in how much weight we gain or how lean we remain, which is, I think, pretty amazing. And the third example I wanted to mention today, just very briefly, is when bacteria take up some chemicals from our food and then turn them into some very exotic molecules, very exotic metabolites, such as hormones and neurotransmitters. And this is very interesting because if you imagine that a bacteria can produce serotonin in your gut, and that this serotonin can be absorbed in your body, it can send signals to your brain, determining your mood. So there is actually a really large area of research that is trying to look at the connection between our guts and the brain. It's called the gut-brain axis. And apparently there are many more ways that bacteria actually communicate with our brains, determining our mood, how hungry we are, or even how aggressive we can behave, and things like that. Some researchers claim even that some mental health issues might be linked to certain impairs in our gut microbiota. So these were the three examples that I wanted to mention to you briefly, and I'm not sure how I'm doing with time.
Maybe we have time for some
All right? Anyone?
What about the antibiotics? When you take that, you just mess up.
Exactly. Basically, when you take antibiotics, then you don't only kill those bacteria that are harming you, but they are killing everyone that is present. And it seems like for some people, it can take over 2 years to recover their gut flora after one antibiotic treatment. And if you get this treatment when you are 1 or 2 years old, then it can have really profound effects, because gut bacteria are supposed to teach your immune system who is friend and who is foe. So those children that had antibiotic treatments or were not exposed to enough environmental microbes, they can have serious risks for developing allergies and autoimmune diseases. Any other questions?
Otherwise, they can die if they don't take antibiotics, then you can choose.
Yes, of course. I mean, I'm not saying that you should not take an antibiotic when you're... Pardon?
That was kind of the message I heard.
Yes, I didn't try to suggest don't take antibiotics if you're attacked by a serious pathogen. By any means, do so.
There's a difference between the broad-spectrum antibiotics that you get over the counter in some parts of the world and the more targeted ones that might be better for specific diseases, right? So that's also part of
Of course. I'm not a specialist in antibiotics, but yes, I could definitely agree.
So just for, if you could go back to the slide with the amount. Is it on? There we go. The amount of microbial cells. There's some a recent review in Nature that stated that this is actually not the case. That well, it depends on how you count cells, of course. Because, for instance, if you count the red blood cells, then it would be a very different picture. But apparently, it's based on research from the 1970s. I was wondering if you've heard about this and sort of how it affects the image.
Yeah, yes, it affects the image, but I think this number has been used for a really long time, and I really believe that it's mainly for the purpose of popularizing this subject, and I agree that it is not necessarily the most accurate estimate, and it will depend from person to person. So I'm not trying to tell you that this is an exact number. I'm just trying to tell you that there is a lot of microbial cells in our body, and that it appears that they are actually important.
But the message is just as strong if there are as many microbial cells as human ones, don't you think?
Yes, I guess so.
Looking at that picture, why do we wash our hands after all?
I guess people have different reasons for washing their hands, but of course we don't want to be completely dirty if you're bending in your garden and you go in for a dinner. It seems that kind of natural just to wash your hand these days, but of course if there's an outbreak of disease, you really want to wash your hands to make sure that you're not carrying pathogens with you. But
May I comment on that?
yes, please.
We have a transient and we have an intrinsic flora, a permanent flora that is supposed to be part of our body, and then we pick up from the environment. So that's the one we want to wash away. And that's why we don't want to use antimicrobials on our hands. We want to use soap, basically, because then we don't touch our permanent flora. We remove our transient flora. So that's a very important point to pick up here.
Yes, sure. But it also depends on the soap that
So
you're
please
using.
wash your hands after you pee and shit.
All right, I guess I'm gaining a really bad reputation here tonight, but never mind that.
I think we should give Andrea a good hand.
Thank you.
Thank you. And we have something for you. You have the. all our presenters are given a little token of gratitude, actually a trophy, saying that you've been the proud presenter of Art, Science and Cocktail.
Thank you very much.
Thank you so much. So next one up is gonna get ready. She's gonna get her mic on, so we might want to give her a couple of minutes. But it's going to continue the kind of discussion from Andrea with microbes and bacteria, because we have the assistant of the artist Gins Gabrans with us here, Anna Babre, who is working for RIXI, Institute of Technology in Latvia. So Gins Gabrans is one of our artists presenting his work Food at the Article Biennial, where he's been working with microbes that we actually have in our gut. And under the notion that we cannot process the cellulose that good, like a lot of the vegetables that we eat are not even processed, and they just disappear out of our body without us being able to take in all the nutrients. So Gins Gabrans has been gene manipulating this enzyme and kind of making a super enzyme that can process the cellulose a lot better, even to the extent that we would be able to eat grass, bark, paper, or or cardboard. So I really would like you to come and have a look at the exhibition, art exhibition for Article Biennial, to see Gins Gabrans' work. And today Anna Babre will give us a presentation of Gins. Anna, please.
Good evening, everyone. It looks like Hege has already done my job presenting Gins, but I will try to come after that. So, already as mentioned, my name is Anna. I'm working for RIXI Center of New Media Culture, Riga, Latvia. And we are art producing company of, we have artists, we produce different events, and etc. But today I'm here to present Gins' work, Foods, that you could see yesterday at gallery, and he's sitting there right in the corner, so you can say hi to him later maybe. So about the work, as Hege already mentioned, it's a transgenic bacteria of a human metabolism that breaks down the cellulose. RIXI and Gins have cooperated in a lot of projects in past for several years where he has exhibited this work in different versions. So first you can see pictures from our exhibition Fields, which took place 2 years ago in Riga during the year when Riga was culture capital of Europe 2014. And this is how his work looked there. At that exhibition, we tried to combine different fields that are usually perhaps not that combined, like the artistics with the social and the nature, the scientific and the emotional, as well as the sensible with the actual. The next project, as you can see, one again picture, was Cafe Europe, which we created together with Mons 2015, which was culture capital a year later. So again, this took place in 2014. Here you can see a building process. This is Speaker Quartier, whereas our office, it's one of the biggest creative quartiers in Riga, in center. And this is how the Cafe Europe looked. We had each week one like specific theme, which we tried to explain to students, to just random people who come and visited us. And this is the picture of the work. You can see the soldier in exhibition down gallery as well. And yeah, as I mentioned, we have student groups too who just visited us, and we have like some workshops with biologists and etc. This is our exhibition in Mons last year, and we exhibited not only Gins' works, but our work Talk to Me, where we worked with the plants and try to make them talk to us and us talk to them. And then later we brought this exhibition to Riga during our festival last year in October, where Hege as well participated with her work, Human Fuel. And now a little bit more about this work, and let's go to the numbers. So in the first century, all over the planet, there was only 150 million of people. in the start of the 19th century, there was already 978 million people. And United Nations organization is predicting that to 2050, there will be 9 billion people all over the planet. So it's a pretty huge difference in numbers. And it's already now hard to feed all the people. So if we go to the next slide, do some of you have any idea what could be these numbers? Sorry? No. This is data from World Health Organization that says that every day 46,000 people die from hunger all over the world. So if we separate them to smaller parts, it means that every hour more than 1,500 people die. Every minute there's 28 people, and each 2 seconds there's 1 people who die from hunger. So what our artists from Latvia, Gins, and his colleagues from Biology Center are trying to do, they're trying to bring up the idea that perhaps we don't need these 70 extra % of food production that we would need until that's 2050, but that we can use our body to just degrading the cellulose enzymes. As you can see, there are some most popular foods linked in the table. So if we could have this enzyme, the difference between perhaps potato calories on 100 grams wouldn't be this much. It's like 9 calories more. But we could use other food resources marked in yellow, like perhaps bread and paper. If we now can get 0.1 calorie from paper, 100 grams in paper, then later we could get up to 400 calories. So basically, as some people like to say, we could eat books and be smarter, not just from brain, but perhaps from other resources. And here's one of ways how Gaines shows his works. It's the clay statues that he makes. Well, not he, but he with his colleagues. And it's one of the works. So here we can see the wood. This small piece is with us without this bacteria, and the big chunk is if we could if we have this enzyme. And what's interesting that the scientists are not the only one who's interested in these type of projects. In 2004, the United States Defense Department launched a competition where they tried to encourage scientists to make, let's call him super soldier, that could just walk on fields, and he wouldn't need any food resources carrying with him or something like that. He could just survive from air. So this one could be a good project for this competition. Unfortunately, it closed before Gaines came up with this idea. But yeah, basically, if soldier is somewhere in an environment which has grass, paper, wood, he would just be fed, and he doesn't have to bring huge bags with the provision with him. And here we can see some pictures how perhaps our future dinner will look. This is a pretty nice dessert, I think. As I like to say, these are made for French people, so yeah. And because I have an opportunity, I would like to invite you all to our festival in 29th of September. It takes place in Latvia National Library. We will have more than 500 square meters of artworks dedicated to art and science, and we will have a conference as well with such people as Christian Paul from New School in New York and UC Parika. Well, that's all. Thank you.
Maybe you can wait till I run over with the microphone.
The first question is, how exactly do we just eat these bacteria and we get the enzyme? What happens? We're not going to modify ourselves with the gene, right?
See, I have to get the precise answer for that. tu saprati to jautājumu, kā mēs iegūsim, bet mēs to baktēriju apēdīsim, un neteicu, ka mēs to...
Varbūt tās... labāk tulko. Ka tas... īstenībā tādas baktērijas es negribētu teikt, kādā ceļā ievada. Organizmā.
Bet, vai ir bet?
Nē, bet tas nav domāts kā galējs risinājums. Es nezinu, kas būtu, ja tiešām tādas baktērijas cilvēkam būtu.
So basically, he wouldn't like to say the way how could we get these bacteria in which way, but it's a different solution because we haven't created like this type of bacteria or enzyme now. So I think if it could be possible, then we could figure this out later how to like put it in our bodies.
Un iespējams es neatrisinātu kaut kādu nākotnes pārtiks trūkumu pārdzīvotā pasaulē. Ja iespējams tas novest pie cita apokalipstīgas scenārija.
Yeah, so basically, and it's not like 100% solution because it can provide other apocalyptic scenarios like diabetes, perhaps, because like the calories are filled with the sugar at one point, so it's not like ideal perspective.
There was another question. I'll hand you the microphone.
Okay, so we'll just skip the problem about how we generate this enzyme, okay? But let's take for granted that somehow we do generate this enzyme. What about the terrible deforestation of the planet when we start eating all the trees?
Pieņemsim, ka mēs izgudrojam šo te baktēriju, kas notiks ar to, kad visi koki pazudīs no zemes virsmas tajā brīdī.
Pirmām kārtām tās baktērijas, kas tajos attēlos, viņas ir pa īstam ārpus ķermeņa ģenētiski modificētas. Tā kā straks pagaidām vēl nav noticis. Kas būtu nākotnē, nezinu.
Well, the fact is that they are already modifies that outside our body and that there is nothing bad happened yet, thank God. But you'll never know what will be in the future, so he doesn't have a specific answer for this one.
One more question.
Yes, thanks a lot for your presentation. I have a question regarding your reason for focusing so much on paper, which is already a processed material and contains chemicals that can't be processed. Whereas I can follow you when you say we can eat grass or wood, but I am curious about your position about paper and the other elements in paper that are not cellulose. Thanks.
Par to, ka papīrā ir daudz ķimikālijas, un kāpēc tu fokusējies tieši uz papīrā? Papīrā ir tik daudz ķimikālijas, un teorētiski viņas nonāk organismā. Ja viņi piekrīt par zāli, kokiem un tam līdzīgi, tad papīrs ir tāds.
Bet papīrā arī visvairāk celulozes, tāpēc, ka papīrs ir visvairāk apkārt. Tā fokusēšanās ir tāpēc, ka tai grafikā bija papīrs, it kā tīrs papīrs ir pilnīgi no celulozes, un viņā ir visvairāk tās kilokalorijas. Tāpēc vairāk nekā kokā zālē.
Well, he focused on paper because there's more like the cellulose than in grass or the woods, and he thinks about the clean paper, not like the chemical improval.
Thanks a lot.
Thank you.
Give Anna and Gins a hand, please. And I guess you will be the one to receive this.
Thank you.
Thanks a lot.
Thank you.
So now we will have a quick break, like 10-15 minutes, so you are able to top up your drinks and explore our wonderful cocktail menu. Shortly back. It seems like the queue to the bar is getting smaller, so which means we should probably start with our program. If someone is missing chairs, there are some extra chairs in the back here. It's really nice to see so many people coming in. Next presenter is Dr. Christian Thorsen. He's a professor at Bioinformatics, Department of Electrical Engineering and Computer Science at the Stavanger University. He will be presenting the tendency to prevent change, control and regulation as a condition for life. Welcome.
Thank you. Okay. First, I would just like to thank for the invitation to come here and talk to you. So today I'm going to talk about how the ability that organisms have to create their own internal environment and to regulate this environment, how this ability is very vital for life to exist. So all types of organisms, from the simplest bacteria to multicellular organisms like ourselves, have a protective border around them, so that they, inside this border, can have the conditions that are needed to run all the biochemical processes, all the chemical reactions that actually make out life. So, for instance, to have your DNA copied, to have metabolisms going on, to produce proteins, you need to have a certain concentration of different chemicals. And this has to be a very quite narrow range where these processes actually can happen. So for the simplest bacteria or the simplest cell, you just have a cell wall, which you can just think of as a very viscous layer, kind of oily, like a soap bubble. Of course, some cells might have tougher walls, and multicellular organisms and complex organisms might have specialized cells whose only job is to create this protective layer, like the bark of a tree or our skin. So like our skin cells, their primary job is to protect the cells on the inside of our body. And to talk a little bit of why it's important to create an internal environment and what kind of advantages that provides, I'm going to first talk about something a bit higher up than the chemicals and the concentration of chemicals. I'm going to talk about temperature regulation in higher animals and in ourselves. So in some way, this works in the same way by keeping our inside at a constant temperature, regardless of how the temperature is outside, and regardless of how the temperature changes. It makes us much more agile, it makes us movable, it makes it all of the processes inside us can run at an optimal all the time. So we have a better ability to react to danger, for example. And also it makes us much more adaptable. We can adapt to a multitude of climates. We don't necessarily have to be in the sun in order to have our processes in the body work. And of course, not all animals have this ability, but those who have it have an advantage. So the modern scientific understanding of regulatory mechanisms and how this works in organisms was set forth in the 1920s by Walter B. Cannon. So he introduced this concept which is called homeostasis. So he used the word homeo, which is kind of in the same range, so not exactly at the same set point all the time, but in narrow limits, and stasis for staying in that range. But of course, scientists before him were also pointing out that we have an internal environment that is fixed, that is at a certain constant level compared to the outside. But Cannon thought more about the regulatory mechanisms. What kind of mechanisms are going into creating this environment? And thinking about it in a bit more an engineering way, comparing this to like how your body would regulate temperature to how an engineer would regulate the temperature in a building. So we have mechanisms to increase temperature, and we have mechanisms to decrease it. And in the same way, right, if I'm too hot, I will start to sweat, and heat will start evaporating to control my inner temperature. The same way, if I'm cold, I will start to shiver to create body heat. So Cannon, he did a lot of animal experiments, and some of the animals that he experimented with were cats. So this particular cat, he did an experiment on to try to get hold of where exactly does thermoregulation happen in our body. Is it delocalized to all our cells? Does it have to do with our brain? Or how is this connected? So we have two nerves running from our back of our brain along our spine, and we're sending signals from our brain and out to our body. And this cat have had this nerve thread removed on one side. So on one side, he operated on it, removed the nerve thread, patched it all together, and of course, after recuperated after the operation, the cat was put in a cold room. And this is the response that we see. So we see that its hairs are risen on one side of the body, but none on the other. So the thermoregulatory system of the cat is lifting the hairs to increase isolation, to provide better isolation. But we see here that it only works on half of the cat. So in some way, this tells us that there has to be a centralized control of this. That this probably happens somewhere in the brain, and then signals are sent out from the brain to the cells that actually tells them you have to extend the hairs. So we can still see that the cat is sensing that it is too cold, because it's functioning on one side, and also that shows that it's sensing that it's too cold, is that these cats also shiver. If it's cold enough, they start to shiver. And they shiver over their whole body, not only on one side. And that also tells us something about that these two different mechanisms for controlling inner temperature are not totally connected. So you could have a totally fine functioning systems for creating body heat, which does not depend on this nerve and this connection to your brain, but a system that provides better insulation does. So, of course, I work with related issues. I don't work with cats. I don't work with higher animals. I work with yeast. The same simple unicellular organisms that are responsible for the alcohol in your drinks tonight. Some of the most important organisms we know. And the reason to work with regulation and control and how it works is, of course, that a lot of the diseases and disorders that we have are really just malfunctioning regulatory systems in our body. So, for instance, if you have diabetes, it's really just a breakdown of the regulatory system that will control your blood glucose level, so the level of sugar in your blood. So if a healthy person like me, if I eat sugar, the level of sugar in my blood will increase, but then my body will detect that and start to produce insulin. Insulin promotes the removal of sugar from my bloodstream into the liver and into the cell, and there's this nice negative feedback causing the blood sugar to drop again to a healthy level. If you're diabetic, you might produce little insulin or no insulin at all, so if you eat a lot of sugar, your blood sugar will just continue to rise, and there's no mechanism in your body to actually remove excess of sugar, so you just continue to skyrocket to unhealthy levels. And then you might experience that you have to be your own regulator, so you can take insulin shots. So you're taking over the regulatory job that your body should actually do itself. I'm working on a much more basic level than trying to cure a particular disease. I'm working more on exactly how does the system work, how can we manipulate them, and what kind of mechanisms is it possible to make or is it possible to have. And this is what I do in yeast. So one of the systems I've looked on is the regulation of copper in yeast. So copper is one of these trace elements that we need some of, but too much of it is toxic, like many things. So copper is actually used a lot in agriculture as a way of stopping microorganism growth in general. So you don't want yeast or mold or bacteria or whatnot to grow on your agriculture products, so you spray them with pesticides or you spray them with copper. And you get this bluish copper color here from the leaves is from copper sulfate salt. And what happens then is that these coppers enter the yeast cell, and it disrupts the functions inside the cell, and it dies. So I was thinking, or what this project goes around to do is actually the opposite of what we thought we would do. I was thinking, can we make yeast more resistant to copper? We would probably think, could we make it less resistant so that we don't have to use as much? But we can't, there's no use of that really, because I can't control what kind of organisms are in the wild anyway, in nature. I can't try to insert a gene and hope that that will survive. So I would rather do some basic science, not that applications in mind, and just try, okay, can I create some sort of super yeast? Just to see if I can. It's a neat system. So basically what this system consists of is 2 parts of DNA. So there's one part of DNA that acts as a sensor. It detects copper, and when it detects copper, it turns on the gene that follows. So the DNA that's coded after it. So this is called a promoter. So when there is copper nearby, it turns on the next gene, following it in the DNA code. So the next gene in the DNA code is actually a gene that codes for a protein that sticks to the cell membrane and pumps copper out of the cell. It actually uses ATP, or energy, in the yeast cell to do this, but you are actually able to remove copper. So you get this system with a nice negative feedback. If there's copper in the cell, this switch, or this promoter turns on, you start producing the pump, and the pump removes copper. So you have copper, you start removing it. And in that way, you give these cells the ability to control their inner environment. These cells didn't have that ability to control it for copper, so we're giving it to them. And then the last graph on the right there is just some results showing what happens with the modified yeast cells to the left, the non-modified yeast cells to the right, and you have the columns in different shades of gray, and the lighter shade is a higher concentration of copper. And the height of the columns is how many times the cell population doubles over 50 hours. So for each doubling, I have twice the amount of cells in my colony. And what we see is that both the unmodified and the modified cells, for the 2 lowest concentrations, they're both happy. They're living, they're multiplying. But once we increases it, we see that the unmodified cells, they die off, and the non-modified, or the modified cells, they continue to grow. They don't grow as fast as they normally would, but they still are able to live with this and to grow. So yeah, I think it's a neat system, although I don't have much applications in mind for it. It's more figuring out, can we do this, and does it work? So with that, thank you.
Does anyone have any questions? Yes.
Thank you. It is a neat system. Well done. When you say you gave the yeast this ability to pump copper out, where did you get it from? Where did
that's also an interesting question. So this protein is, of course, from another species. We're not come so far that we can kind of create our own proteins de novo just for typing the sequence and knowing what kind of function it will have. So we have to take it from somewhere. And this is actually taken from a different related species, which is known as Candida, which is an infective yeast. Some of you might be familiar with it. And this has this copper pump, and it's thought that this is also one of the reasons why it is able to live inside a body, because we have an internal environment that would create more loose copper ions to go into a yeast cell who would live inside a body. So they have created this through evolution. Whereas the normal yeast cells that are used for baking bread or making beer or alcohol do not have it.
Any other questions? Yes.
Yeah, you say that this is basic science and that you don't have many applications in mind, but of course it's always interesting to speculate, right? And hearing your description, I'm thinking like when you can't control the yeast in an external environment and you have a mechanism for killing that yeast. If you introduce this super yeast, you could use the mechanism and leave only the yeast that you have created.
I wouldn't go into those speculations in this talk, but of course we thought about it, and of course because getting funding in science today is about pointing out applications, of course. And that's just one thing that we've been thinking about, is that we could create an environment where we're very sure that this is the only yeast that's living here. So we have very control of what kind of fermentation is going on or something like that. The only problem there is copper in itself, because it's something that for food stuff you wouldn't want to add it to when you're brewing, for example, to make sure that you only have one type of yeast, because you would contaminate your beer with too much copper ions. But it could be a nice way in general for selection also in lab work, if you could replace that and use that inside as a marker or something.
What form of copper is exported by this protein?
So it exports copper 1+ ions. And this is the only kind of copper ion that is normally inside the yeast cell. So you normally have copper 1+, copper 2+. You have reductases in the yeast cells and on the surface of the yeast cells that will take copper 2+ and reduces to copper 1+. So most of the internal copper is in the form of copper 1+. Yeah.
Okay, thank you. And here's your trophy.
Thanks.
So our next presenter is going to be Margarita Pevere. She is also one of the artists for the article biennial, where she is presenting her work Seminina Eternitatis. But she's an artist and independent researcher, and she's working with audiovisual work, organic reliquaries, paperwork, bacterial culture, and gene manipulation. And today she's going to talk to us about beyond transiency of life, living matter, decay, and memory as artistic media. Welcome, Margarita.
Thanks. I'm sorry, I forgot the remote control. Where is it? Oh, it's here. Thanks. Didn't see it. Yes, thanks a lot, everybody, for being here. I would like to thank Hege and Nora to organize such an extraordinary exhibition, and I would like also to thank my fellow artists for bringing such inspiring works, and Kulturhaus Stavanger for hosting us, and you as an audience for being here and sharing some thoughts with us tonight. As Hege introduced, I'm presenting my artistic research, which I will continue during a PhD at the University of Aalto in Finland, where I will focus on transience using living matter decay processes, and is inspired by memory. I brought you a picture and a quote as an inspiration for you tonight. The picture is a picture that I took of the skeleton of a lizard, which unfortunately I killed while playing with my dogs. I moved a lot as the dogs were chasing lizard, and I moved a lot to set it free, but instead of setting it free, I smashed its skull. And I was very sad for that, because I didn't mean to do that. And I decided to wait and see what happened to this lizard, and I came back at the end of the summer, and that was the result. So I could observe how the nature and microorganisms and insects ate the flesh and left only the skeleton, which is, for me, very beautiful. It looks like a membrane, and I still have it in my collection of bones. And the quote is by Wilhelm Flusser. For those who are not familiar with his work, he's one of the most luminous mind of post-war generation of intellectual. He's a media theorist, but he somehow also anticipated some instances of feminism and of the Anthropocene, and he worked a lot as an art critic. And this quote says, mankind is different from all other known beings by the fact that it acquires information, stores it, processes it, and transmits it to future generations. Mankind is anti-natural in the sense that it is committed against the entropy of nature. So there is an action that humans carry on this planet that is against entropy of nature, and it's maybe an attempt to control the erosive process of time. Who knows if that works? We will see in the future. And I found this quote in the Wilhelm Flusser archive in Berlin, which is the city where I live. And it's open. If you happen to be in Berlin, please go and visit the UDK. They will be happy to show the original papers of Flusser. Now, I will tell you a bit about my artistic practice by showing you three works that are related to each other. One of these is exhibited in the Stavanger Kulturhus. I would say that my artistic practice is based on a series of counterpoints on manipulation and erasure, preservation and decay, evolution and erosion. And I do this with technology, and I do this also with living matter and organic matter. We'll see first this series of works. It's works on paper. For this, I use a very simple technology, as I use only paper, a glue that I make, and pencils, so graphite and maybe China ink. So I use plain cellulose, a semantic handwriting, which is a way of writing that is not meant to convey a meaning, but rather to show the gesture of writing and leaving a trace. A semantic writing is a definition by Gillo Dolfles, an Italian art critic. And in this series of work, I focus on the evocative power of words, on the physical gesture of handwriting, and on the resemblance of cellulose with body tissues, which I investigate on an aesthetic level. And in fact, the works have no frame at all. They rely only on the materiality of cellulose. You might ask, where's the technology in it? Or where's the science in it? And I would reply maybe, yeah, can we speak about technology and science and our relation to it without using it directly? Or can we make a step back and then evoke its function and how we relate to this? The next work is more related to technology and science in the sense that I've been manipulating 8mm, 16mm, and 35mm film strips. It's all found footage, so don't worry, I have not disrupted any valuable artwork. And I've been manipulating it with the help of my own microbiome, as I use my own gastric juice, of my gastric enzymes. I've been held by sprouts and mold for the 35mm, and I've been erasing and rewriting, as I did in this series of work where you see the words that are fading. I did the same on the 16mm film strip. So on this, I focus on the erosion of the visual information stored in the gelatin layer. You might know that the photographic film has two layers. One is cellulose, plant cellulose. Well, it's cellulose acetate, which have not been disrupted by any of my actions. Whereas the second layer, which is gelatin, and in this case animal gelatin, and this is the layer that contains the information, has been corroded by my action. You see here, that's the result of my gastric juice on the 8mm film strip, and that's the result of molds and sprouts on 35mm. You see, these are the audio strips. Again, the molds and sprouts only acted on the gelatin layer and not on the cellulose layer. Now we come to the work that I am presenting at the article Biennial. This picture has been taken by the Stavanger Abendblatt, and who kindly sent me over to me. In this work, I push further my use of biotechnology, in the sense that I am collecting memories of strangers, of people that I don't know personally, and I work with a bacterial culture. It's a cellulose culture of Glucoacetobacter, and the aim of the project is to store the collection of memories in form of digital data transformed into DNA sequence into the DNA of the cellulose, so that I can grow cellulose with human memories embedded into it. In this work, I focus on the cross-pollination of visual art and synthetic or digital, as my colleague Rudiger Trojok, the German biohacker, say. So synthetical biology, hybridity, and blurred notion of species. In this sense, it's also inspired by the presence of a microbiome by the concept that we are not only human, and so which Andrea, where's Andrea? Here, which she mentioned so interestingly during her talk. Now you see that's a page, or that's a layer, a sheet of cellulose. It's still fresh, and with this cellulose that I grow in a tray system, I am making the artist book. I've made the artist book that you can see here. The brownish page is made by this exactly material. And this is a culture that was usually adopted for turning wine into vinegar. So it was actually very present in the everyday experience of the past generation, whereas now when we go to the supermarket to buy vinegar, we forgot that we can have our own culture at home, helping us, providing us with food. So as you can see, I recombine living matter with that technology. I thank Hege for suggesting me this combination of words in the investigation of permanence and transience. And I would like to specify that I use memory and the term memory for its double reference, human memory of your own personal memories and memory in terms of technological storage of data, as a reference to the passing of time. So there is no autobiography in my work, at least not mine. You won't find any traces of my own history in my works. And I should point out also that working with living matter and decay processes brought me to face a sense of objections, as living matter can decay, and the decay processes are not always very nice to see or to smell. And you need to learn how to manipulate them in order to obtain an artwork. But this can become part of the artistic process, like when I work with my own gastric juice to manipulate the film strips. So I just embodied that in my own work. And before saying goodbye, I would like to say a couple of words about the New Eden, which is the title of this edition of the article Biennial. As an artist, I can't be but excited about the new possibilities that are coming up with the new scientific findings and new technology, and also with the democratization of this technology. But at the same time, as an artist, I can't prompt a critical understanding or wishing for a critical understanding of how we relate both to the technology that we are using, but also on how we affect the biosphere. So how does this biotechnology redefine the human position in the biosphere? And I wish there will be more exhibitions like this to discuss about this theme. So, thank you.
Thanks so much. Is there any questions from Margarita? Yes.
In the film strips, do you keep all of the results, or do you just select certain ones?
I kept all. I worked in this way first, since it's found footage, so I was not focusing on the exact content of the pictures, like in the works on paper. I'm not focusing on the meaning of each word. I cut some strips and I made some tests, and then when I decided how to intervene on every specific film strip, I enacted the strategy. But I can show them to you. I have some here with me, so it can be a topic for discussion.
Hi, I was just curious, does the way, when you insert these sequences into the DNA, does it affect the way the DNA functions? Or? Yeah.
Thanks. That's a good question. I am not sure yet about what technology I will adopt, as I am handling right now with a few labs for doing that. So it will depend a bit. The technology will depend upon the scientific team I will collaborate with. And the idea is to insert DNA which is not meaningful to the living organism. And in this way, I am aware we might encounter the risk that this DNA is discarded, but it might also stay latent. And that's a question mark, and this is part of the research project indeed, because it's not my intention to modify the organism, rather to give an extra or augmented to a certain extent an augmented DNA.
We have techniques for that.
Well, let's speak about that.
Well, I'd like to thank you, Margarita. And here's the trophy for you.
I love it. Thanks.
So now I think we will have a short break again, about 10 tops 15 minutes. If you want to refill your glass, go ahead. Okay. Then we are getting ready for the last round of presenters. And the first presenter, oops, was that me? The first presenter in this round is Dr. Christian Ballot. Professor Christian is he's a professor at Intelligent Information Access, Department of Electrical Engineering and Computer Science at the university. He will be presenting artificial intelligence from search engines to digital personal assistants. Welcome, Christian.
Yes, good evening. Hi. I think you can hear me. I was told to be entertaining, so I hope everyone had his and her second or third glass of whatever you are drinking. So I want to talk to you about some of the intelligent systems and technology that surrounds us, and you use these on a daily basis. And I would like to put this in a somewhat bigger perspective of artificial intelligence. Now, to talk about this, first we would need to define what we mean by intelligence. And there are many different definitions around. This is the one I use, the ability to learn, understand, reason, and solve problems quickly and effectively. Remembering things is not among them, so I can just read it from here. Artificial intelligence is when these things are performed by a machine. And in most science fiction movies, already 50, 60 years ago, this is portrayed as a computer, a box that people can make an intelligent conversation with. And you can see just some examples here, and some are more human-like, some are not human-like at all, just boxes. Okay, so my field is in search engines, and in very plain terms, these are just computer programs that are designed to find information. And what is interesting about them is that the search engines are one of the most sophisticated artificial intelligence systems that exist today. If I had to name the second one, it would probably be self-driving cars, but that is far, far behind. So when I say search engines, most people think of Google immediately, and yes, Google is one of the biggest players, but there are also other search engines, Facebook, Amazon, YouTube, and they have similarly intelligent and advanced search. Okay, so just to give you a very high-level idea how it works, there's a user who types something into the search box. We call it a search query. This is sent to the search engine, the computer program, and that program responds with some search results. And so far, there is, of course, it has to happen very quickly and efficiently, so that is intelligent about it, but that's not the end of the story. What many people don't know is when you click on something, then this information is sent back to the search engine, and the search engine learns from this feedback and tries to improve itself. So the more you use a search engine, the better results you get. So for example, if you are interested in cats and you search for jaguar, then you will get results about the cat. If you are more of a guy and interested in cars, then if you type in jaguar, you will get results about the car. Okay, so this can be seen as a mission statement for our field. What we are concerned with is making the right information available to the right people at the right time. This is a picture I see very often at my university, but I also see it at the train station, or on a bus, or an airport, or in many bars. People engaged with their smart devices. And this is a big game changer for us, because search is no longer just performed on a PC or laptop, but it's with you everywhere. And since you take these devices with you, we know a lot more about you. And that's why there is a transition from the search engines into personal digital assistants. And it stands on 3 pillars that I will talk a little bit about, or illustrate them with examples. The ability to answer, to converse, and to anticipate. This is something that you already see a lot on Google, for example. Search is no longer just a ranked list of documents, but it tries to give you direct answers. So if you search for the books by a given author, then you can get a result page like this. If you search for the weather, it knows that where you live. It even knows my postcode, because I was logged in. If you ask how to tie a tie, then you get the instructions with pictures. You can ask for Olympic medal count, and then it figures out that you are interested in the current Olympic Games. And you can even ask about the earnings of some celebrity. And in all these cases, you get this knowledge panel or card that gives you a direct answer. Okay. The second point is conversation. This is something that I actually performed earlier today with Siri on my iPhone. I asked it, what is the capital of Norway? Well, I had to repeat it like 3 times for it to get my pronunciation, but eventually this is how it was transcribed. What is important that what is behind is just a search engine. This voice interface just translates your voice into a written search query, and then it responds with a card. And in a follow-up question, I would ask, okay, what is its population? And then it knows the context. It knows that I'm talking about referring to Oslo, and it responds appropriately. So that's very nice. And what is also nice about this conversational interface is if it's unsure about how to interpret a request, it can ask for clarification. So if you forgot to update your contacts in your iPhone and you want to call your husband, then it lets you choose which husband you want. Okay, the third one is anticipation. In this scenario, we can see that someone is traveling, and this is Google Now. It brings up a reminder that should leave home at some point. Then the flight was cancelled. It gives a notification, and immediately after that offers alternatives for finding a different flight. Let's say that this person managed to travel eventually, then it brings up the boarding pass when she arrives at the airport, and when she lands, then the system shows the weather and interesting places to visit in London. So these personal digital assistants can be imagined like these virtual secretaries, and one of the things secretaries do is they update your calendar and send you reminders. So this is actually real examples from my computer. When I made a flight booking, then it immediately put it in my calendar. It reads my email. It understands that this is a flight booking. And it also gave me a reminder that I should leave 9:30 today, and the traffic should be light. Okay. So what is the future? Going back to the movie example, when it's about AI, then it's often portrayed something like this. These intelligent systems will destroy the world. The question I want to ask is, how far are we from real intelligence? And I want to answer this with a simple imaginary scenario. So let's say that these kids are playing with marbles outside on the street. And I ask the question, what is 3 times 4? And the first kid answers 7, which is the right answer to a different question. Then the second one answers 12. Okay, great. Why? And then he says, I don't know. And then the little girl aligns these 12 marbles like this, and then I know that she understands why 12 is the correct answer. So at this point, these search engines are very much like the first and the second kid. In many cases, they give the right answer without knowing exactly why, and in other cases, they just recognize that 3 and 4 appear together, so it might be 7. I would like to close with this slide, just going back to the original definition of intelligence, and use this to highlight what I think are the biggest challenges for AI. So understanding and reasoning both have to do with being able to provide an explanation and an evidence. So when there's an answer, the system should be able to explain why that is, why it believes that is a good answer. The second one is we are good in, even better than humans, on very specific, very narrow problems. But general purpose AI that does well on all sorts of problems, I think is well beyond our reach. Thank you.
Thank you. Do you have any questions from the audience already? Yes.
I have one, and thanks for the interesting presentation. My question is, so you focus on artificial intelligence for algorithms and search algorithms, or do you apply that also to other fields? Thanks.
Mostly search, but in the broadest sense that that I had on the slide. So making the right information available to the right people at the right time. So
by the way, it's not only because it's an extremely wide field. This one.
There's 2 more questions. I'll start with the first one over here.
Is there a difference between the? Okay. Hello. Is there a difference between machine learning and artificial intelligence? And maybe it's a 2-part, but and where would you put Watson in prospection of your presentation?
Yes. So it depends on who you ask. Artificial intelligence is broader than machine learning. Machine learning it can be seen as a subfield of artificial intelligence. Natural language processing can also be seen as a subfield of artificial intelligence. And my field, which is called information retrieval, is also a subfield of artificial intelligence, which combines many things and borrows techniques from machine learning and natural language processing. Yes, the second question, Watson, that's a question answering system. So that could be, well, it's not a personal digital assistant, but in a way an intelligent search system that was programmed to do well on a very specific quiz. But this falls under our area of interest.
I have a question about language in the conversation. There was a series of questions and answers. What is the capital of Norway? Oslo is the capital of Norway. What is its population? The answer was given for Oslo, but the reference is to Norway. Why did it answer for Oslo rather than Norway?
To me, if you asked me, I would have answered the question the same way, because I follow up on the answer to the previous question. But indeed, the third question could have been, okay, what about Norway? And then it should still understand that we are talking about population.
Right. I was just curious because in writing, you're never supposed to do that.
What do you mean?
When you use a pronoun, it refers to the last noun, which was Norway.
No, the last noun of the answer was Oslo. But that's an excellent example why conversational interfaces are great. Because when there is a confusion or ambiguity, then you can just explain it to the system instead of formulating it as a very long query and typing it. Yeah.
I have a question about ambient. An ambience of a system is in touch, feel, smell, see, hear. And so when you have an artificial intelligence, the ability, according to your definition of intelligence, a system able to feel its way through or smell its way through or taste its way through. And the question is, which is the superior system? Or, I mean, can artificial intelligence reach that level, that ambient level?
I'm not sure I fully get the question, but my answer would be you can program a system to feel and smell, but I don't know if it could...
Okay, the question is that the way you take the children, the way one learns is through tactile, right? It's touch, tactile. It's tactile, right? So the problem with our cell phones or our problem with our pads is that actually you can see a flower but you can't smell it. Sometimes you can Google flower, rose. You get a rose, but you can't really feel the thorn, smell the rose, etc. Right? So is this a positive or a negative according to future systems?
I think that's one of the biggest limitations, is the system can do anything that you program it to do. So if you make these inputs available as signals, then you can program it to behave in a certain way. But on its own, it won't it won't miss the smell, or it won't miss the colors.
Well, interesting questions there at the end, and I think it also kind of gives us a notion of how utterly complex we humans are. So I have a trophy for you.
Thank you
Thanks
very
a
much.
bunch.
Thank you very much.
So now our last presenter is going to get ready. We are so lucky to have a presenter that's come here all the way from Tromsø today. It's Dr. Fern Wixon. She also has been writing a text in our catalog. Dr. Fern Wixon, she's a senior scientist and program coordinator of the Society, Ecology and Ethics Department at Genøk for Biosafety in Tromsø. She will be presenting here for us today. Is there a place for GMOs in the Noah's Ark for seeds? Welcome.
Thank you. Thank you. I want to also start by thanking the organizers. It's a fantastic event, and I'm seriously impressed. I've tried to run science cafes myself in Tromsø and never had half this amount of people. So really, congratulations on an It's the cocktails. I'll remember that for next time for sure. So I'm here, and I'm going to talk to you a little bit about a project called the Biodiversity Project that I've been coordinating now for a bit over a year. The project is about agricultural biodiversity and the importance of agricultural biodiversity. We need it, this is talking about the biodiversity of our crop plants, so plants like maize and wheat and soy, the diversity of types that are actually available in the world around those. And that's considered really crucial for future food security in the sense of how are we going to adapt to new challenges, new pests, new diseases, new climate change. We need diversity in our crop plants to be able to draw on and adapt them to these coming situations. So there's a general recognition that we need crop biodiversity for future food security. So the Biodiversity Project's really interested in, so how do we protect that? How do we look after lots of different types of maize plants and make sure we keep them around? And there's basically two models for how we do that in the world. One is, well, you get farmers to keep doing what farmers have always been doing, which is growing lots of different types of crops and continuing to experiment with them and breeding them and on the farm adapting them to different climates, different conditions, different places. So one way is in situ, in context conservation. The other way we seek to protect this biodiversity is we put it in a freezer, in a gene bank. We take it off the farm in a seed form and we freeze it down so that researchers now and in the future can experiment with it, use it and play with it and make new varieties that way. So this project's interested in both of those angles and we're working with indigenous farmers in Mexico to help them conserve their biodiversity, and we're using art to help do that actually. But today I want to talk to you about the other form, which is the freezing down in the gene bank form. And I want to talk specifically about the Noah's Ark of seeds. Does anyone know what I mean when I say the Noah's Ark of seeds? Have you heard that before? Exactly. The Svalbard Global Seed Vault, which is right there. And I want to read, so I was there for the project researching, and I want to read you some field notes from when I was there, when I was standing outside these doors. After that, I've written some notes to try and give you a sense of what it's like in this place. So I've written, I stood at the base of a frozen mountain on an island high up in the Arctic archipelago. The wind was tearing through me in icy shards as snowdrift whipped around my ankles. Nestling my hands deeper into my pockets, I scanned the landscape. An airport, a port, a stain of abandoned coal mines, a scattering of houses against a backdrop of steeply rising white mountains and deeply plunging blue seas, ocean and ice, snow and stone, raw rugged nature and doggedly hardy humanity. I'd come to a place where polar bears outnumber people and is dark for months at a time. Here I stood before a locked metal door leading deep into the heart of a mountain. In there, said my guide, is the most biodiverse room in the world. The contrast of that, of standing in this freezing, icy, bare, naked landscape and being told that I was about to enter the most biodiverse room in the world was weird. And it really is the most biodiverse room in the world because it houses currently 860,000 samples of 5,000 different species of plants alone from over 230 different countries. That's astounding. And basically, when you go into the vault, you enter these doors, and it's literally a long tunnel that is deep into the mountain, and then there's 3 caverns carved out. And in those caverns are boxes and boxes and boxes of seeds. It's a very strange place. So it basically, what its purpose is to hold backup of the collections in gene banks around the world. So all across the world there are gene banks that have their freezers full of seeds, and Svalbard is a backup, a duplicate copy. The idea being that gene banks are kind of vulnerable. There's all kinds of things that can go wrong with them. You can have a power failure and your freezers stop. You could have a natural disaster and floods wash out your collection. Or you could have socio-political unrest, war that destroys your collection. So there was an idea internationally that really to make these seeds and varieties safe, we need a backup. We need a place where the collections can store their backup. And it took a long time for everyone to agree that this was possible. but in 2008 they opened the Vault. It's run by a kind of triplicate of organizations. The Norwegian government is kind of the owner, and they built it. They took the bill for building it as well. Then there's an international NGO, the Crop Trust, that fundraises to get money to enable the whole place to run. And then the practicality of accepting the boxes and putting the boxes in the vault is done by the Nordic Genetic Resource Centre, Nordgen. It's really important that I let you know that it's a black box system. So a lot of people are worried about who has access to the seeds, who can get them out of there. Is it Monsanto, for example, that somehow has access to these seeds? It is only the depositing institute that is allowed to withdraw the seeds. So basically, a gene bank from anywhere in the world puts their seeds in a box, the box gets sealed, shipped to Svalbard, someone collects it at Svalbard, and then they carry it up and they put it on a shelf. No one is allowed to open that box. Only the person who deposited it can make a request to get that box back. That's only happened one time in the whole existence of the vault, and that was recently by some Syrian researchers who couldn't access their gene bank in Aleppo anymore, and they said, could we please have our box so we can work on the seeds in Egypt? So further from my field notes, so I went there to study how the vault works. I was going to be there for some weeks and do some in-depth field work to really understand how it works. And it took about an hour. We went to the airport, we picked up the box, we put the box on the shelf, and that was it. So my field notes after that were, the door is locked, the boxes are sealed, nobody gets in, nobody works here. Sealed shipments are collected at the airport and transferred to the shelves. And there they can sit for all eternity, frozen. There's no testing, no checking, no real work being done. There's no hum of workers, no practice to observe. The hallway is empty and still, bound by concrete and ice. There's no smell of the soil, no touch of the sun, no sound of the rain. Sealed boxes of seeds sitting on shelves in a freezer. That's it. The cavern echoes in silence. Can this really be the most biodiverse room in the world? So it's fascinating for me to be there and think about this is how we're protecting our seeds and protecting our biodiversity. Now, I work in a centre that looks at biotechnology, genetically modified organism. We're interested in the risks that they might pose to the environment. So it was quite natural for me while I was there to ask, so you've got all these seeds, you've got 860,000 samples. Are there any GMOs in the vault? Genetically modified crops, do we back them up and store them as well? And I got really weird answers. I got lots of different answers from those 3 organizations that look after it. Some were saying, no, I don't think so. Some were saying, yeah, sure, why not? And so I had to really dig a bit deeper. Eventually, I started to get answers converging in 3 forms. No, there's no GMOs in the vault. The vault is not a GM-certified facility, meaning if you want to work with genetically modified crops, you have to get kind of approval that your facilities are good enough to work with them, mainly to contain them so they don't get out. So you have to have non-porous building materials, netting on your windows, all this kind of thing. They said, no, we don't have any because the vault is not certified to take GMOs. It's a very good answer. But of course, they could be if they wanted to be, because of course they're very contained. They're buried deep in a mountain in Svalbard. Those seeds getting out and surviving in the wild, quite unlikely. Then the second type of answer I got was, no, there's no political will. Both the government and the current managers of the vault were like, you know what, GMOs, kind of a controversial topic, trying to avoid it. So actively accepting them into the vault is not something we're willing to do. There's no political will to do that. They also said that GM crops don't meet the conditions for multilateral access. This is interesting in the sense that any depositor wanting to put their seeds in the vault should have signed an international treaty around agricultural genetic resources that says they should be open for sharing. And if I have these seeds and you want them, I should be able to share them with you. If you want to develop a great new variety and make $1 million out of it, you're welcome to, but you should then share the benefits of what you've done. So the people who've signed onto this treaty, which is most of the countries of the world, are sharing their genetic resources in this sense, and therefore they can deposit in the vault. But of course, GMOs are not the common heritage of humanity like the rest of the seeds of the world. GMOs have been granted patented property rights. The people who've made GMOs, they get monopoly ownership rights because it's no longer deemed to be a living organism. It's an invention, and an invention can be patented. And then the point of a patent is not to share, right? So GMOs are actually very different from the rest of the seeds that live in the vault. I just wanted to say that there is the potential for this to change in the sense the government told me they could get a waiver for the certification. If they wanted to, they could do that and they could store GMOs in the vault. You could also have change politically. Suddenly there's political will, or maybe the gene banks depositing suddenly want to put GMOs in the vault, so it could change. There's also possibilities to get exemptions around multilateral access conditions. So the vault also has written in its contract, if it wants to make an exemption, it can. But there's also the potential for contamination. So GMOs contaminate other kinds of crops because they cross-pollinate through the pollen sharing that happens naturally in the field. So of course, trans genes can spread into different crop varieties, and then they could be, if you're not testing and you're not checking, deposited in the vault unknowingly. And of course, the vault isn't checking, and they're not requiring their depositors to check. So there could be contamination. And then I started thinking, well, the most interesting question is perhaps not, are they in there? But rather, should they be in there? Do genetically modified organisms have conservation value? Because here I was sitting with a project that was saying, we value crop biodiversity. This is one of the most important resources in the world. We really value it. And almost everybody agrees with that. Now, when it comes to the value of a genetically modified crop, no one is advocating that. No one is standing up and saying, oh, they have such important value, we really need to protect them. They're so dear to us and so cherished. I didn't find anyone talking about that. So I tried to then understand, okay, so do they have conservation value? What would be the arguments for and against that? And I would love to continue that conversation with everyone over cocktails, because I have some ideas, but I'd love to hear what other people think about this issue. So the first thing that came up was you really have to think carefully about how or why are they significantly different from traditionally bred crops. So very often people will say, no, 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 a GMO is not valuable because, you know, a human has made that. It's not natural. Very common kind of go-to response. But then no crop is natural. Every single crop that we eat, crop plants, they're always a co-creation between humanity and the plant. That's the way it is. So then you say, what has really changed with the GMO? And why, and in a sense, what has changed so much that it's lost its value in terms of being conservation worthy or its moral status? So you have to think, really come up with a good argument for that, and I have yet to do that. You've got people who say, maybe they add to biodiversity. What you've done with the GMO is taken bits of genes and genetic code from different organisms, viruses and bacteria, and put them into a plant that couldn't have got there naturally, which is why you can patent it. So wow, you've kind of created a radical new form of biodiversity. This is, you know, adding to the biodiversity of the planet. Why aren't we all kind of really excited about that and thinking that's protection worthy? Some people will say they have conservation value in principle. Okay, so we don't know what's going to happen in the future. We should be protecting all kinds of diversity just in case we might need it. So yes, in principle they have value. But maybe we can serve them differently in practice. Maybe they shouldn't be in gene banks. Maybe they're in the field. So typically we've thought of the old traditional varieties are the ones we look after in the field with the farmers, and it's the modern varieties that we have in the gene banks. But I'm seeing a switch in which it's the modern varieties that are spreading out into the fields, overtaking the areas dedicated to traditional varieties, and now the gene banks are saying, we better take and protect the traditional varieties because they're disappearing from the fields. So maybe they're conserved differently. You've also got the idea that maybe they have instrumental value, they could be useful for us, sure, that's one of the big claims when people are trying to sell you a GMO, it's going to be useful for feeding the future. So maybe we could acknowledge they may have instrumental value, but you could say they don't have intrinsic value, or they don't have what some people call integrity value, which is a value often given to wild biodiversity, genetic integrity idea, that they don't have that. But then you could say how, on what basis, really give me the deep philosophical argument for how that happened. And indeed, with a GMO, you could say you're intentionally violating integrity value. You're intentionally violating integrity. So if anyone here has a strong position or idea about whether GMOs have conservation value or not, and it doesn't sort of fall into these kinds of argument lines, please let me know and I'll add it to the research project. When I was doing this, though, I really realized that I think the whole debate is getting really messy and we need some new terminology to help us reimagine things in new ways. Particularly now as genetic technologies are diversifying rapidly. The things we've known before about transgenic GMOs are not the future. We've got genome editing, CRISPR and Cas9 systems which are taking off and creating all kinds of different organisms. And we need to think carefully about what is the moral status of those organisms that we're making and what is their conservation value. So I was thinking about why don't we talk about biodiversity? We talk about synthetic biology, but what about the products of that as a form of biodiversity? And if we use the idea of biodiversity, you could think of all those different types of biotechnological organisms or artistically created organisms that come up, and this could be a category term that covers them. The term GMO is very kind of linked to the type of GMOs that we have today. So to try and use that same term to apply to all the ones that are coming tomorrow is going to be difficult. So I'm thinking maybe we want to start talking about biodiversity. But then the problem with that for me is that the bit, the synthetic bit, puts a little bit too much weight on the human and a little bit too less weight on the plant. I mean, we're collaborating in this initiative, really. So the biodiversity is the idea of symbiosis, where two organisms cooperate for mutual benefit. It's a biological concept. So maybe we need to be thinking about when we're creating all these organisms, is it biodiversity? Is it mutually beneficial? Because under biodiversity, you could include both biotechnological plants and the traditional varieties. They would both belong under that biodiversity. And I think we really need to start thinking about, do they have conservation value, but also how do we care for them? We think a lot about whether we should or should not do technological interventions and what might be the risks and how should we regulate it. But we don't think so much about how to care for the organisms that we're creating on the other end of this process. And this is where art can be really powerful and really useful to help us stimulate our reflection about, oh, is this something that we should be taking care of? And how do we relate to this organism as an organism, as opposed to an invention as such? So this is a very famous and quite old work that is very good at sponsoring ethical reflection around biotech development. But what I'm missing is the application to plants. Because here, yes, we would all kind of immediately feel a need to discuss ethically this kind of entity. But if I said this was a genetically modified entity, which it is, where is the emotional connection to the stimulation of the discussion? So I would really like to see us have a really nice chat about, is there a place for GMOs in the Noah's Ark of seeds? And that's what I had to say today.
Well, I'm sure this is gonna start a whole avalanche of questions. So we can start with a couple of questions, and then I suggest we continue the conversation over the tables, with cocktails.
And I'm going to say, every time I present on GMOs, I'm used to being attacked. But interestingly, I work at a biosafety institute, right? So I'm normally seen as an anti-GMO activist. So today I get to see what it's like if I don't take a very strong anti-GMO role and see if I get attacked in the same way. Let's go.
Okay.
Okay, I want to be careful to phrase these as questions.
No need, you can make statements.
Well, on Symbio, I was wondering about companion species. We go to all this trouble to hold this enormous biological record, but I would assume that many of these plants cannot exist without significant companion species, and are they also preserved?
No, so this is one of the weirdest things about the idea of conserving agricultural biodiversity in the freezer. You remove it completely from its ecological context, completely. When you look at, they have what they call passport data. So if you take out a seed and you say, oh, what's this seed about? Where does it come from? How does it grow? What does it grow together with? You know, what is the ecosystem you need to make this thing function? The passport data has none of that. The passport data literally says where it was collected, by who, maybe a GPS coordinate kind of thing. It's extremely divorced from its entire ecological context, and its cultural context, which is part of our project's mission to work with bringing that in.
Also, you made a remark about how the difference between GMO plants and non-GMO plants in their commercial context, as GMOs are usually patented, but aren't many non-GMO plants patented as well?
No, so only the US has a Plant Patent Act, and even then it's very restricted to what kinds of plants you can patent, and even then the patent you have is not the same as a patent given to a GMO. A GMO patent is a utility patent, so you get plant breeders' rights with traditional varieties. So you get some kind of ownership right, and you get the right to benefit. But what the rights don't extend to is research and saving and replanting. So with plant breeders' rights and even plant patents under the US system, you are not allowed to prohibit research to be done, and you're not allowed to say you can't save that seed. You're not allowed to sell what you save, but that's a different thing. Whereas GMOs, the utility patents they're awarded say, you are not allowed to do any research and you're not allowed to save your seed. So it's an extra layer on top of the property rights that are awarded.
In the case of many non-GMO garden vegetable seeds, they're hybrids. You can only buy them once because you can't replant them. You have to go buy them again from this.
You can replant them. You won't
but you get the same
output.
Right. So
but that's a different. Yeah. So in practice, you could say that's the case. But in property rights, it's not the same, because with a hybrid, you can replant, you won't get the same output. You can also research it. So we've got farmers in Mexico, for example, who they do replant hybrids, and they do experimentally cross the hybrids with their own maize. So they have that freedom, not that they get the same consistent results. But certainly in a modern system like the US, growing only hybrids, the difference is kind of slight.
Okay, one last question. I won't hold the floor after this. I would like to talk to you later. I have been collaborating with the United States Department of Agriculture's apple collection, and where they have thousands of apples species, and they do not preserve GMO apple species. They refuse to have anything to do with GMO apple species. But
do they say why?
You know, I think your point about the political atmosphere may have something to do with it. But another thing is that species have been disappearing long before GMO species hit the dirt. I mean, there are like heirloom apples that people go hunting around for in the woods that disappeared 150 years ago.
Absolutely.
So the fact that species are disappearing isn't solely the responsibility of GMO producers, it would seem. Do you agree?
I don't think I said that, but yeah, for sure, species are disappearing way before GMOs. I think what you're seeing is possibly an acceleration, but it's also not GMO related. We're seeing usually it's a modern agricultural package that is leading to the disappearance of traditional varieties, and GMOs are often one part of that package, but not necessarily. Certainly in Mexico, they haven't approved GMOs for cultivation, and that they're losing massive amounts of traditional varieties of maize, but that's because of the adoption of modern hybrid agriculture, and GMOs are just an extension and an expansion on that.
Thank you for a very compelling talk.
Then we have another question.
Yeah, my question is that we have a seed bank now. Do we need a soil bank? Because you have a seed. I mean, because you can store all the seed you want, and in 1,000 years you go back and they won't grow in the soil because it won't. Okay, so what are we going to do about that?
Yeah. Look, to be honest, people talk all the time about the gene bank and the in situ, in-farm kind of conservation models are complementary. You need both. They both need to exist. But what we see is a massive amount of difference in the financial investment. Gene banks, they're not even seed banks anymore. They're gene banks. We're conserving genes, not even seeds. That's getting all our money, while the idea of keeping farming diverse is not being supported. It's expected to just happen. It's expected that, you should be supporting. And what we're seeing with the Mexican... Yeah, so they're saying they're complementary, but if they're truly complementary, then I would hope that they're getting the same level of investment, the same level of care, the same level of protection. And what we see in our system is that there's a massive support for gene banks, but the idea of keeping things alive on the farm in diverse... Totally. The whole gene bank idea is kind of crazy in a sense. And it only works because it's a gene bank, because you have this idea that if you conserve the genes, then you've got all the information that you need. Because, of course, it's the soil, it's the companion species, it's knowing the climate, the season, when to plant, what to plant. It's the socio-ecological package that you need to conserve, which we're really worried that as long as we keep going down the gene bank model and it keeps rising and the other one keeps disappearing, you lose all of that. Massive risk.
Okay, I've got 3 more questions from there, and over there. And I will close it off after that, so you can talk to people individually, okay?
Mm-hmm.
This is a simpleton's question. A GMO-modified species, don't they have a recipe for it? Couldn't they make it again? How repeatable is that process of recreating?
Yeah, good question.
And do they then need to have them in the bank if they can just take one there and make it again?
Yeah, so you could say that, and that sounds like a perfect plan. In reality, the way we make GMOs today is a random insertion process. That's improving with new techniques. But so you just kind of shoot a gene in, and it will land anywhere in the genome. And so typically, you will have hundreds of modified seeds, say, and maybe if you're lucky, one of them will have the trait that you want and no negative features. So it's actually really hard to get it to work perfectly. That's improving with new techniques, but the way we do it now, it's been extraordinarily difficult to find that right one. It's often one in thousands of trials. Yeah, although they say that it's faster in a sense, because you're selecting you don't have to grow the whole plant necessarily, but CRISPR is making a big difference. CRISPR is being more precise, and that's why I say it's really important that we don't get locked in the idea of a GMO being what we know today, because CRISPR is changing the world and changing the types of organisms that we're going to be generating.
So let's go a question over here.
Yeah, fascinating talk, thank you. I was lucky enough to be at the inauguration of the Global Seed Vault in 2008, and I actually even carried a box of chicken pea into the vault. So it was kind of amazing. And so I talked to a couple of people there. At that time, I was running a project on agrobiodiversity, and we were wondering if this is really a great contribution to the conservation of agrobiodiversity, because there's so many places around the world where they collect the seeds and freeze the seeds as well, and not just one. There are several places where they have the same seeds. So actually, the situation where this wall will actually have a real benefit or can save the situation. This is a really very special type of mid to large global catastrophe where this can have a say. But the real benefit that I think was that this was the biggest kind of public relation activity on the importance of agrobiodiversity that was ever taking place. At that time, we were doing a documentary, and we put the clip online on Wikipedia to tell the people about it. I think it was the clip the most watched we ever produced. But it was in all the media all over the world reporting about it, and everybody was talking about why it's important that there is agrobiodiversity. What is it? Why is it important? And the same people that actually were driving the inauguration and the building of the seed vault I think they're coming from the conservation community, which is traditionally anti-GMO. That's very clear because of this interruption of the Plant Variety Protection Act and the way that the plant breeders share the seeds, and which is now interrupted because there are patents, a different kind of intellectual property rights. But I think they should be in there because the world is planned for centuries, maybe even millennia to last, and the patent only lasts for 20 years.
Yeah.
So to put it in there, wait 20 years, and there's no more patents, you can share it whatever you want. And this is just the beginning, because the next thing that's going to come is, we were talking about CRISPR, we're talking about the total expansion of biodiversity beyond the chemistry that we know right now.
Yeah,
there are different types of nucleic acids that can be used, and it goes way beyond what we see right now in nature. So we should be more open to include all kinds of and symbio or xenobiodiversity.
Yeah. Yeah. But I would say a few things to that. I would say that GMOs are patented right now, but they don't have to be. You know, there are research institutes who are saying they won't claim a patent, and then they missed that kind of reason for not being in there. The Vault is hugely popular. It's in video games, it's in crime novels, it's in all kinds of weird places. It has this kind of mystique around it, you know, this hole in the earth of the mountain in the Arctic holding all the world's seeds. It's kind of magical. But it's important to realize that it's not meant to help the world survive in a global disaster. It is a backup for gene banks. So even if the world went under and we lost all our crop plants, we would still not be able to access the stuff in the Vault, and that's not what its purpose for is either. So it's a bit of a strange organization, but it's very interesting.
So we're gonna stand knocking at the door, hungry?
You won't even get to the door.
Oh my goodness. One last question,
Go
then
ahead.
we'll let you loose.
Yeah, maybe a stupid question, but is there any kind of problem actually by, now there was 860,000 different seeds stored. Is there any storage problem why you won't allow GMO? Is that any kind of problem? Why can't you store everything, just dig more larger holes? Svalbard is very big. So I don't understand the basic problem of your speech.
It is not a storage problem. They actually, when they built it, they made 3 caverns, like rooms slightly bigger than this. 3 caverns. Only one of those is currently in use, and it isn't full yet. But the idea is they've got 2 more massive storage rooms for all the seeds that are still going to keep being delivered there. So there is certainly plenty of room for the GMOs. Yeah. Thank you very much.
Thank you. And here's your trophy. Thanks a lot. You've deserved it. And just a reminder for you to go and see the new Eden. I'll put the flyers on the table so you can have some information. Oh, yes, we have a panel debate tomorrow. We're going to have 3 of the other artists on our program that's going to present tomorrow. It starts at 1 o'clock at the gallery space, Sølvberget, at the Culture House. So from... Yeah, so the panel debate is going to be from 1 o'clock to 4 o'clock. And if any of you want to have a guided tour in the exhibition before the panel, you can join us at 12 at the gallery, okay? And thanks a bunch for coming. I've really, really enjoyed having you all here tonight. Thanks a bunch.